Is it on?
Yeah. I failed the IQ test.
All right. Is it on? Okay. Well, thank you very much, John, for the introduction, and thank you very much for giving me the opportunity to present here at this wonderful Cornell University. And if you did not attend the great ACERT workshop, be certain you guys have this great ESR facility at site. Now, as John said, I'm going to present the application of biomolecular ESR, and specifically DR spectroscopy, in which case the relation is to measure molecular distances of proteins and therefore correlate the underlying structure towards its biological function. Now, before we continue, why do we want to use ESR for answering biochemical question? Now, part of the reason is that in general, proteins do not have paramagnetics. Yes, there are metalloproteins, but in general, that's not the case. And therefore, it is very easy to introduce paramagnetics at the believable site of interest and probe, therefore, its biological structure, equilibrium, and eventually kinetics. Now, furthermore, ESR is not the end of all things. Indeed, it is just part of the big biophysical toolbox. Therefore, integrating it with NMR, with MD calculation is key. However, it does answer key questions, which is unapproachable to many other methods, such as amyloids, for example, or membrane protein, because in this case scenario, indeed, they're just too big for NMR, or we might not be able to crystallography that out. Furthermore, due to its characteristics of freezing sample, we can sample along the energy landscape of this protein and look at variety of heterogeneous system. Now, what do we look at when we look at a protein? If we specifically look at specific labeling site where we introduce this observable, this paramagnetic label, it looks something like here as we see it. Therefore, the paramagnetic label is rather small, and indeed it is the size of a residue and subsequently minimally disturbing the ultimate protein structure. Just for your own sake, if you compare that to a fluorophore, yeah, the MTSR, the paramagnetic label is rather smaller. What can we do? We can measure distances, specifically double electron resonance spectroscopy. We measure distance between 15 to 160 angstroms, so 1 to 60 nanometers. We can look directly, because it's a direct measure, it's a dipolar coupling, rather than looking at indirect methods. Furthermore, we have small label. We freeze samples. That means we can freeze out substates. We can look at individual samples, and we can do in vivo measurements. But I want to be certain we cannot get atomic resolution, at least in the case of DR spectroscopy. How do we approach normally a biological problem, a biochemical problem? Let's say we have two domains, a red domain and a blue domain, and they're arranged randomly in space. And really the answer is what we're looking for is how are they arranged? Therefore, restraining them with one single distance could answer our question. Now, to go ahead, we will choose a paramagnetic labeling site as shown here and introduce our paramagnetic label. Upon doing so twice, we now can measure a paramagnetic distance in space that will ultimately give us a dipolar evolution time trace that subsequently can now, thanks to mathematical manipulation, extract a distance population that subsequently can be fed into our initial problem and therefore restrain ultimately the domain arrangement and solve our question of what is the underlying structure to cause this biological function. Now, here I'm going to have 3 case studies. One of them is simply to look at the structural arrangement. This is a very straightforward problem. We ask, what is the structure? Subsequently, we're going to look into a GPCR, because GPCRs are great. They are primary drug target of many pharmaceutical companies, and they are very important in our daily life. And subsequently, at last, we look at the crown jewel. We look at protein kinetics. which is very hard to get. And I will convince you indeed ESR can do such thing. Now let's look first at HIV. HIV is like a disease which needs no introduction. We all know, we're all aware how it normally works is the virus gains entry into the cell, after which viral RNA is transcribed by reverse transcriptase towards proviral DNA that ultimately can be integrated into the host genome. Now, you will notice that really one of the key points is reverse transcriptase. If we find a way to stop reverse transcriptase, we will block viral replication. Great. Now, furthermore interesting is, actually, HIV reverse transcriptase initially exists in a monomeric state that during the maturation process will form the homodimer that is biologically not functional. And then subsequently, upon cleavage with HIV protease, it will form its biological functional heterodimer. Interestingly, this homodimer means they're identical subunits. They're identical sequence, okay? Now, the question what we are asking is, what is the structure, and can we target the structure with existing drugs? Do those drugs, those antivirals, work on the homodimer? And furthermore, how does HIV protease get access to the cleaved domain, the RNase domain, to ultimately result in a biologically functional reverse transcriptase that will cause viral replication? So to start up, we need to make mutants. As I said, we need to find mutation sites where we will attach our paramagnetic label. We need to know around what distances we're interested in. So as we see right here, this is our mutation library. This is around 40, results in about 40 distances. So therefore, it's a large number. Then we can characterize them pretty much because it's a homodimer in intermolecular distances. So distances with across the subunits, or we're going to look at intramolecular distances where this is within the individual subunit. Furthermore, we're also going to look at intermolecular distances which are mixed, where they can cross each other, okay? Those are three different categories what we're going to take into. So meaning that we're going to get a dipolar time trace shown in blue. And we also, just for fun, we're also going to add a drug, a known antiviral that actually works on the heterodimer itself. So this is shown in red. And as you can see, blue and red in most cases looks identical. However, in some cases, it actually changes. Therefore, it already shows that indeed the drug interacts with the homodimer. Now, upon acquiring those dipolar evolution time traces, we can now translate them over into distances. On those distances, indeed, we can now map into our HIV reverse transcriptase. We can feed those dipolar time traces into our structural calculation and subsequently gain an understanding how the HIV reverse transcriptase homodimer looks like. And furthermore, because we also did it with the presence of a drug, we can look how the drug-bound state looks like as well. So we get the price for the price of one. And what you're going to see here, there is around 20 structures overlaid after the calculation. We can simply see, indeed, actually the HIV reverse transcriptase homodimer, that means both sequences are identical, or is fully asymmetrical. Furthermore, upon drug binding, it has a similar effect as seen in the crystal structures, where it opens up this single domain, which is very important to clamp onto the RNA and subsequently allow for the translation from RNA or the transcription of RNA to DNA. Now, what I'm not showing here is actually the RNase domain. The RNase domain, this is the domain which ultimately gets removed by the protease to permit the translation into its biological functional form, is presented by two distance distribution, 1 and 2. That is, this one will give rise to two-member ensemble where one of them, one RNase domain, is actually collapsed onto the homodimer, whereby the state 2, is rather extended. Now, you will already understand right away that indeed, probably the extended state allows for some access and ultimately results in the cleavage of the RNase-H domain. Now, this is correct. Indeed, we did this experiment where we used our HIV resistant homodimer, added our protease, and subsequently this subunit was removed, and we resulted into that biological functional heterodimer. That can be easily seen when you look with protease. It's a very flat line there because it removed the RNase-H domain. Okay. Now, I told you from the side of reverse transcriptase, but we can also go the other way. We can actually now go back and look of how HIV protease does its biological function. So this is exactly what we did in this case scenario now. We took one-time HIV protease that will cleave the RNase-H subunit. In this case, is inactive. Once added to it, you can see that indeed it will stabilize the state 2, the elongated RNase-H domain, because it will clamp onto it and will stabilize the elongated state. Furthermore, if we now use paramagnetic labeling scheme to observe HIV protease directly in action, you can see that in the free state, you have a broad distance that ultimately, upon clamping onto HIV prior to actually performing its proteolytic function, will result into a very longer distances, because those MTSL labels, those labels which we are seeing, they actually act for the space filling model. That means if HIV protease will bind to its cleavage site, it will be those labels will be displaced and therefore have a longer distance. So now what we have seen is, yes, indeed, we have developed an understanding of how the HIV reverse transcriptase homodimer looks like. But not just that, we didn't stop there. We were curious about the maturation, and we now understand that the two-member assembly, so two-domain orientation, will allow HIV protease to clamp onto the linker connecting the RNase-H domain and cause a proteolytic cleavage, subsequently giving rise to the HIV reverse transcriptase heterodimer and its biological function. Now, this was published in PNAS subsequently under many other publications, but the nice thing when you know it's doing all right is if somebody writes a report. In this case, David Weber wrote a nice article about our article saying an asymmetry that leads to activity. So this was very much highlighted. Now, before continuing, does anybody have a quick question? Otherwise, we do it later. Okay. So we're going to shift from structure towards equilibrium study. We're going to shift from a viral protein, from a reverse transcriptase, towards a GPCR, and going to look at how drug interaction will occur and then ultimately result into GPCR homodimer formation. Now, GPCRs are obviously very important. They regulate a large amount of our biochemical functionality. In this case, we're going to look into the beta-1 adrenic receptor. This beta-1 adrenic receptor is placed, for example, onto your heart and makes your heart beat at a specific rhythm. Now, this regulation is occurring partly due to a dimerization event. For example, when the lipid environment is correct, or in the presence of cholesterol, it will bind towards the GPCR. If it's, for example, in the presence of similar detergents or lipids like cholate, subsequently, this will actually further result in dimerization of that GPCR, okay, and therefore regulating its biological function. Now, this was in collaboration with Nia Kobatova, and what you can see is that the question what we're trying to ask, what can we do or how can we explain the regularization from monomer towards a dimeric state of the GPCR, okay? In doing so, we attached two paramagnetic labels and initially just recorded the GPCR in the sterile-free case inside a micelle environment. Now, this is what you can see. You can see a distance signature of 3 distances. Now, that result, that explains that there's nothing wrong, it's just a GPCR. Now, subsequently, we're going to add cholesterol, or CHS, which is a mimic of cholesterol. And upon doing so, you will see that those 3 distances actually collapse into 2 distances. It's because they actually bind. There's actually a binding event where GPC or beta-1 adrenic receptor is present, and we have CHS actually bound. Subsequently, we're going to do the same thing, and now we're going from our sterile-free case into binding to cholate. Upon the interaction with cholate, we can now see a dimerization event, which is characterized by a rise of distance around 25 angstrom. Now, of course, we want to characterize this dimerization. We want to characterize that binding event of the of cholesterol. So every time when I think about it, I would automatically think, well, this is asking for a titration, because we're going from one distance distribution to another distance distribution, and we're asking what rate are we going there? What equilibrium constant we have to the binding? And this is exactly what we did. We did where we initially started with our sterile-free GPCR and increased the concentration of our cholesterol mimic, and upon which it binds directly to a binding pocket of the GPCR. And you can see that very nicely with the disappearance of distances over time and the appearance or strengthening of a distance at 35 angstrom. Of course, this is great because now this is just a simple binding equilibrium, meaning we can simply map it out by having a monomeric species interacting with a ligand and therefore indeed calculate the KD. This is very easy to do, and as you can see, those are the fits. Please keep in mind, I never bother fitting to a distance domain, always a time domain, because this is a raw data. I love raw data. And what you can see here, this is the ultimate fit, and we end up with a KD of 1.4 millimolar. Now, this explains the interaction from a sterile-free case towards my ligand binding, the CHA's case. However, I'm obviously interested in the ultimate dimerization, right? And this is due to cholate, and cholate induces dimerization. So what I'm asking is, how do I go from this distance distribution towards this distance distribution? Please keep in mind, there is an additional distance of 25 angstrom. However, as you will notice, there's also a distance right here. So this is a very unique case because this means at no time ever do I have pure monomer or pure dimer. So therefore, we actually had to use the T1 edited deer experiments initially introduced by Fraser McMillan, in which we now can separate the population of the monomeric species and the dimer species based on the number of units present. So meaning that we get a T1 modulation factor, and upon doing such, we can get the distance shown in red, which corresponds to the monomeric unit shown here, or the percentage population, but also shown in red here, we can get the distance distribution specific to the dimer, okay? Because we didn't have that information. Before, it was always a mix. Now we isolated it. After doing this, we have now a base set. We know how the monomer looks like, how really the monomer distance looks like, and the dimer. And we can now establish an titration with the addition of cholate to see the rate of change of how we can go from the monomer structure, or almost all monomer structure, to a mixture of monomer and dimer. We can now simply fit it to a monomer dimerization model. Of course, it was a little more tricky than that, but in general, we have then a KD of 35 millimolar with a Hill coefficient of 2.4, and that really going to describe the dimerization. Now, what I did not tell you, if I have a monomer and I add cholate, I will form a dimer. Great. But furthermore, if I have a monomer and form dimer with cholate, I can actually break that dimer under the addition of cholesterol of our CHS. So if this is the case, and I know already all the KDs that is there now, now I can actually beautifully arrange a schematic where I connect all those different regulation mechanisms. If I go from monomer to dimer, back to monomer, and fully saturate. This is beautiful because now we can really allude to the basic of biochemistry. We understand fully. But how do I know it's true? Well, the way of how I know it's true is if I plug into my set of equations the concentration of GPCR, the concentration of given cholate, and the concentration of given CHS. So meaning if I literally type in the numbers and I am correct, then my output should be identical to my experimental data without fitting, so no fitting. And this is exactly what we did. Over here, shown in this figure, you can see in blue the experimental distance distribution. This is the signature which corresponds in the presence of 0.6 millimolar of CHS and 37 millimolar of cholate. You will see in blue the signature. In red is my simulated. Please keep in mind, not fitted, simulated trace, and it's identical. And the same holds true if I now vary the CHS and the cholate concentration, I can still get identical readout. Therefore, the truth is in the pudding, and I have indeed solved the regulation mechanism thanks to ESR and specifically geospectroscopy. Now, I have shown you how to go about to utilize geospectroscopy to calculate the structure. I've also explained of how I can use geospectroscopy to elude a regulation mechanism of a GPCR that is fundamental for making Valentine's Day work. But now I'm going to your what I personally consider the crown jewel. This is protein kinetics. Okay? Protein kinetics under so pretty much kinetics under non-equilibrium conditions. Now, in this case scenario, we first want to just separate. We have equilibrium conditions where we have, for example, chimodulin in a funnel, and I can assume multiple structures. When I now look at those funnels, I'm going to take snapshots of individual chimodulin, how they look. However, I do not know how they arrange with each other. They're just there in space. Under, however, non-equilibrium condition, I will now be able to take those snapshots and line them up over time and make out of a simple photography and for entire full-blown Arnold Schwarzenegger style movie. So therefore, I believe we might need to rewrite principle of biochemistry by Leininger, because he explains, well, it goes from here to here. But that's not the bitter truth, is it? How does it go from an entire extended chimodulin model, as shown here or here, towards a collapsed state onto its substrate? So let's start first with the equilibrium condition. So this is just looking down the funnel. In this case, we followed very simple principles initially established by actually Fried himself, by freezing it very fast and then seeing what are the individual snapshots. So let me explain. If I take, for example, an MD calculation, I take a modulin, and now I'm going to let it go on over time. What I will get is a range of ensembles. Those ensembles have to be populated. My goal is to use EPR spectroscopy to populate those ensembles and determine which ones are there in this native state. Therefore, I can now attach paramagnetic labels and simulate the distance distribution for each of those ensembles. So what I'm saying is, each of those ensembles has a set of distance that I can simulate, and this will be my base library. This is my library, which I'm going to subsequently use to fit into my data set. Now, here we come. So this is a set of data set. It's a time domain. I know this is very confusing, but what I really want you to take into account, if I take those individual data sets, I will have a range of distance distribution. They're very messy. They're no good. But if I now take all those distance distribution and now fit it, I utilize my library of ensemble of structures that I consider to be present in the equilibrium condition, I can now fit it, shown in red, a very nice direct representation with close to perfect residuals. That means I capture individual ensemble members in those traces. Now it lasts at least, well, right now I'm telling you, I'm taking MMD calculation, which could be made up because we don't know. And now I'm fitting it to a one-dimensional time trace. That's a little bit vague, isn't it? Now, therefore, I'm trying to convince you that we just did not simply fit it. Yes, we fit it. We have a perfect residual. We can determine the percentage. Now, if I take this set of structures and I perform NMR, this is an entire different biophysical method. If I am correct, I should be able to fit NMR data because this is the representation. In this case, we used paramagnetic relaxation enhancement, and what we gain from it is shown in gray. You see the gray trace? This is my goal. That's what I want. Now, each ensemble member should capture part of the PRE, meaning, for example, number 3 captures this broad distribution right here in gray. As I now go 11, I capture another part in gray. Number 12 captures another one, and on and on. The bitter of truth is indeed we captured each ensemble that we have reproduced in both ESR as well as NMR. Two biophysically very different methods, one in a frozen state, one in a solution state, both with their own flaws. But both of them together ultimately could capture the entire ensemble of chimodulin as placed in the native condition. Now, this is equilibrium condition. How do we go about non-equilibrium condition? Now, non-equilibrium condition, we pretty much had to reinvent the wheel. This is from computation. The structures itself are from computation. However, so therefore, we had to reproduce it through 2 biophysical tools because one would not be enough to convince us, actually. Again, those are just models that fit our results. It does not mean they're ultimate truth, but those ensembles exist.
So do the computations also give the percentage population?
No, they did not. And so it was a reweighting problem. You're correct. Yes, of course. Okay, we had to reinvent the wheel. So what did we do here? Again, our question was, if I have chimodulin and I'm adding my ligand, my binding partner, am I able to capture the steps and the rates as it approaches the collapsed state fully saturated with my ligand? Okay? So to do that, we had to literally collect all developments, what we have performed over that many years, and look one time for signal enhancement. So this one pretty much through duration and other aspects, we had to develop an entire new system for rapid freeze quench based on other papers, and then utilize it to capture this each individual time step. Furthermore, we had to use another dimension. We had to come out with a 3-dimensional analysis. Distance alone doesn't do it anymore. We had to look at relaxation times in correlation to distance. And lastly, of course, we had to come up with a way of further analyzing it in a 2-dimensional SVD fashion, which we will publish ultimately. Now, how do we go about it? Let me just introduce you to the idea. In here, we're going to start with an isolated chimodulin, calcium bound. And we're going to have a binding partner, in this case, myosin light chain kinase, a little subunit. And you can see it's very small, and it will bind, chimodulin will literally snap onto it. Upon doing so, we're going to have those 2 proteins mixed in a mixer. That means very well mixed. and then very fast deposit it on a frozen copper disk or cold copper disk, upon which we layer it out very suddenly. Then, upon doing so, we're going to look at individual time steps between 0 milliseconds all the way to 25 milliseconds. Now, each sample produced will also be recorded based on a relaxation-enhanced Deer spectroscopy. Relaxation filter, simply just a T2 edited Deer. Don't worry about that. More important thing is, what will we get? As we go, we can see initially 1.5 millisecond, we have sudden changes. We have 3 peaks. Now, as time goes on, for example, that early peak decreases, the other one increases over time. Furthermore, we can now look at the relaxation, because just simply at those distances would never be enough. We will never get the resolution. To enhance the resolution, we utilize the TM-edited Deer sequence, and therefore we ultimately performed a 3-dimensional analysis. We united distance distribution. So those are the distance, what you can see here. The transverse relaxation time dimension. So this is the relaxation, how long each distance is observable. And afterwards, of course, the kinetics. This is key. And if we now take all those 3 components together, you must imagine a Cartesian space, we will find that one distance distribution that will unite distance, relaxation, and kinetics in one spot. So this is from Carmodulin. So again, we're going to start with 0 milliseconds. Now, this means there's nothing bound to it yet. We have a beautiful distance distribution, as we've seen before. Now, upon going to the next step, let's say in case of 1.5 milliseconds mixing, we can see a distance distribution change. And then obviously in the final state, we're going to see again a distance distribution change. You can see how those distances change over time. When I say change over time, I'm talking about kinetics. I'm talking about rates. Now, of course, to fit this, we need to go over many iterations of forming a kinetic scheme, a pathway, to look at intermediates. We know they exist, obviously. Now, the easiest part is always going linear. We go from free, substate, substate, final. Does that fit? Do the red dots fit to the linear? No, they don't. Let's go to the next one. Let's look at a simple branch model. Free, bound, goes to one substate, ultimately goes to C. Does that fit? If I look at the red spheres and the black one, you can see it's not going to fit either. Actually, the chi-square gets rather bad. And at last, our ultimate result was a complex branch model, in which case now, Carmodulin in the free state can go into 2 different A star and A state, something as sample B, and go to the final state C. Now, what does that mean based on the molecular structure? Again, we start Carmodulin. It can split over where the N either is the N-terminal domain associating with MLCK or the C-terminal domain. So both of those domains can associate with it. Then one goes into both 2 collapse state, but one state B has to do a rearrangement to ultimately snap into its high affinity state and subsequently allow for this interaction to occur. But this is not enough. This is where we should start. This is only EPR. Here we only look at Carmodulin. Reality, we should look at it from other methods. We should look at the atomic resolution, and therefore we collaborated with the Rob Tico group and did some solid state NMR, especially Jay Kyun Jun, where again we utilize now a label the substate, which has specific residues. And our question was, if we do the same method, freeze them up, can we reproduce this biomolecular process, this kinetic process? And indeed, we can. I'm going to get to it in the next slide. But what I just want you to see, if I look at lysine 5, alanine 8, it's a beautiful fit. Alanine 8, 13, and F17, beautiful fit. They are very nice fits based on the same kinetic model as I have shown previously. Now, at last, it comes down to putting the missing pieces together. This is how we should annotate Lehninger. We go on, calmodulin mixing with our substrate, which can now interact. Either the C terminals from the substrate can interact with the N-terminal domain from calmodulin, or the C-terminal domain of calmodulin can interact with the N-terminal domain. Either way, in case of state where the C-terminal interact, it will rapidly snap into its final state. However, if a star occurs and it interacts with the C-terminal domain, then a rearrangement needs to occur, in which case the C-terminal domain and the N-terminal domain come together. The N-terminal of the substrate will snap over into the C-terminal domain, start forming an alpha helix, but not the C-terminal yet, because this one forms rapidly after snapping into its final state. Now, this is very neat, because I swear by, I don't think there has been any way of doing it otherwise. The combination out of ESR spectroscopy, or DO spectroscopy specifically, the assimilation of distance distribution in correlation to its relaxation, and furthermore, observing order formation in an otherwise unstructured peptides results in the entire mechanism of how calmodulin can truly interact with the substrate. At last, I hope you enjoyed the presentation. I hope I have convinced you that indeed ESR, or geospectroscopy specifically, is a very powerful tool in the biophysical toolbox, as it can observe structural effects and otherwise unresolvable arrangements. It can look at membrane protein and its drug interactions and regulation for that process. And at last, it can look at protein kinetics towards the process of forming a biological functional complex. With that being said, I want to thank everybody for listening. I want to thank all my collaborators over that many years, specifically this guy and this guy. And for that further being said, I'm more excited for your questions.
So you have, what do you do here? You have the, MD,
oh, you mean like in regards to HIV? Yeah. Sure thing. So this is a very good question. So no, we don't do MD. Yeah, we do minimization. This is correct. However, what we're doing is more, there's a program called Explore NIH. So similar to MD, we consider specific forces. We consider, of course, backbone, side chain, rotamers, and chi angles. Now, different from that, though, because it's more of an arrangement. So that means the domains, we don't consider them moving. They already pre-existed. In the case of, nowadays we have AlphaFold. We don't need to worry about that. We care more of how those domains are arranged in order with each other. This is key. Now, to do that, though, you're correct. We had to develop an entire new way of looking at rotamers, because simply said, the old way was not enough. Therefore, we consumed the idea of having rotamers, but rather than caring about all those sidechain, we rejected sidechain. We simply said, we don't know how sidechain look like. We do not know how the MTSL, or the paramagnetics, will interact with those sidechains. And therefore, we only consider in our process the backbone arrangement, C alpha and C beta, and of course some other factors, but that's most of it. And at last, based on that, we can then simulate or calculate the rotamer based on a couple weights, and then simply feed in the time traces and see how it fits. We have calibrated this method, so this one is just simply based on a calibration where we have around, I don't know, like 100, 200 distances, which we feed into it to get a calibration of those rotamers based on a known set. But thanks, this is a very good question. So so this is a very good point. You mean like a jackknife procedure? Not a jack-free procedure, jackknife. So no, we have not done that. That's a very good idea by pretty much fitting it. I didn't do that actually. That's a very good idea. So what you're saying is take our 40 distances, calculate the structure, leave out one distance, calculate it again, and see if we can fit in that distance.
That would be good.
That is a good idea.
I would say that this jack.
Yes, we do a lot of different sides. We create 40 distances from all different sides across the protein. Yes, but that jackknife procedure is a very nice idea. I do appreciate that. Thank you. Any other questions? Yes, please.
Yeah, I want to ask you about about the
Uh-huh.
so again, very impressive that process the protein. So I'm wondering whether there is a special preference in each monomer the protein HT.
Say, so how, if the monomer is actually being cleaved rather than the dimer?
So you see there are there is a dimer, right? Two
Mm.
monomers bind together. So I mean, initially it will be symmetry, but if you consider this enzyme bind to the substrate, then it will be a symmetry binding, right? So I'm wondering whether the cleavage of this asymmetry thing, whether created like a symmetry feature of this.
I really appreciate it. I actually had just this discussion today. And this is great. Thank you for the question. You're correct. If the homodimer would be a symmetrical unit, this would be a huge problem because we would not know, the protease would not know which one to cleave. However, we have shown, thanks to EDF spectroscopy, that indeed this homodimer, and to us, please correct me if I'm wrong, this homodimer is actually asymmetrical. It's totally different. The one subunit is absolutely different. The arrangement is totally different from the other subunit, even though the sequence are identical. Now, this is fascinating because I don't know any other case that is this. I don't know, but please correct me if I'm wrong. So therefore, because it's asymmetrical, one of the subunits is popping out and is being actively cleaved off by HA reverse transcriptase. Sorry, protease. So yes, very good question. Yeah. Yes, so actually, it's asymmetry. Actually, it's asymmetry that leads to activity. Yeah, David Weber did a fine job in that title. Yes, please.
Yeah. Very interesting. Yeah.
Thank you.
I was wondering why a paramagnetic, how you know a paramagnetic indicator doesn't affect.
This is a good question, isn't it? Let's be honest, it does probably. I mean, if I would lie if it doesn't. Normally, so this indicator or the paramagnetic label itself, we did not actually pick those mutation sites and randomly. Those mutation sites were already picked based on NMR data. This PRE study, what I've shown, is pretty much just a repetition of a similar PRE study previously done. However, they didn't obviously do a great job in analyzing the data as we did. But in such a manner, they have, however, shown based on ITC and other biophysical tools, so other binding tools or binding methods, that the KD difference that only gets minutely disturbed based on the labeling. And this is a very good point what you're making. And I didn't make much fuss about it, but if you make a mutation and you do add a paramagnetic label, be certain to check for the biological function, because you don't know if that label or the mutation will disturb it. In the case of HA reverse transcriptase, I told you we have 50 distances. I probably made like 80 mutations. And probably 50% were absolute failure because it did not transcribe the RNA to DNA anymore. Okay? So just keep that in mind. Thanks. Very good question. Please. There's a lot to ask. Yes. Yes, Boris. The moon. This is a very good question. I'm going to be very honest. I don't know 100%. And however, I believe because they're just coupled and then you just have flip-flops occurring. So it's just a simple spectral diffusion might do the trick. I'm not 100% certain, but it does. That's correct. But if the paramagnetic label with the dipolar coupled electrons in space are still flip-flopping at a given rate, then they can still affect the T1. Very good question. Thank you. Yes.
So, for example, you change the physics, right?
Of course.
Copper one of that.
Yeah.
How would it affect that? And by how much?
This is a very good question. If I go ahead and take instead of a nitroxide label, now switch over to another paramagnetic label, because obviously this is not the only ones. For example, if I do switch over to copper, so there is, sorry guys. So what Madhu is asking is, currently in the studies, what I presented, I mostly had a nitroxide label, a rather small paramagnetic label, species present. Now, of course, the paramagnetic species or the label itself has many ability, right? So in this case, for example, it's just very commonly used, it works very well, and it's very heavily studied. What Madhu is asking, well, what if I take, instead of a nitroxide label, and let's say I attach a gadolinium label. Now this one, for example, one of the effects is that it's rather big and bulgy compared to our nitroxide. So this is a size comparison. And so is a tridal. But one very nice label, which is commonly used, as I have seen in the ACERT workshop, is the copper label. In this case now, I introduced two histidines, which can cap on individual copper, and therefore introduce a very precise distance. However, I do need to make two mutations there instead of one. So what Madhu is getting at is there are a lot of paramagnetic labels, and all of them have a specific characteristic and a specific use, and therefore it heavily depends on your question, or your biochemical question, what you're asking. If I want to know a very precise structure, then I probably want to have a very sharp distance as shown with a copper label. If I want to detect individual substates or interaction states, I don't want the copper because I'm looking for a space. I'm trying to see what is going on in the surrounding space, in which case mostly I probably want an MTSL or even longer label because I'm looking for the rate of change. If I want to work, as Timothy has shown beautifully, if I want to work in cells, the nitroxide label mostly gets reduced. It's totally useless. However, as I've learned, there's a beautiful flavin label, which does the job very nicely, or as commonly used, a gadolinium, or here we have something called the spider label. So each label, similar to fluorescence, has its specific uses and abuses. So again, it comes down to the specific question you're asking to find that precise answer. Okay, thank you. Other questions? I know there should be many.
I just keep coming back to this homodimer. How does nature break symmetry?
So this is a very good question. You know what? Sadly there was a problem, and I would have loved to actually continue the study. However, turns out HIV funding gets a little bit shortened nowadays. And one thing which I also was asked during one of the meetings today is, well, how about the monomer? Well, how does a monomer actually associate? So how does a monomer go to the heterodimer? I mean, this would be a beautiful kinetic study, actually, right? Because now maybe there is a symmetric homodimer that has a rearrangement for some reason. I don't know.
Just some way with the first one fluctuates, that it tells the second one to sit still, right?
Maybe it's just more like an arrangement of domains. You first have like only two domains maybe interacting, and then the other one falls across it, rather than thinking of an association towards homodimer, rather a scaffolding event. Because what I didn't go much into it, to be fair, but the arrangement of, well, that's what it is right now. The arrangement of this heterodimer, the biological functional heterodimer, actually there is something called the P51 subunit and the P66 subunit. Now, the P51 subunit, the shorter subunit, it's truly a scaffolding. It's literally just there that all the other domains stay in place put. And this is an intriguing little thing. Maybe HIV developed that way because there are other reverse transcriptases as well available, which are having other formations. So I do not know the answer, honestly, but I think it would be interesting to eventually continue the studies if funding becomes available.
Look at the bottom control.
No, I didn't actually. No, actually, there's something else. So we developed a nice method called the IMD, inversion modulated spectroscopy method. And in it, it turns out that there's always dimer present. You almost can't get rid of it. I can't be able, actually, even with the presence of glycerol and all this stuff, so I can't do it. But it would be interesting to maybe think about other ways, for example, through mutation and other methods. Oh, thank you.
So when I was in
You did, didn't you?
a minor
Yes. The flaps.
things up like plane. So then a bond that type plane has bond or group in that same subset. So this intrinsic
Mm.
so it could be something as simple as that, to form an interaction.
Yes, absolutely. Maybe just an initial hydrogen bonding causes the asymmetry, like one little pebble down the mudslide. Just a little side note, since we talked, I didn't go much into it. And I'm actually this is kind of an exciting side fact, since we're talking about your studies with Dennis Torsha on HIV protease. This is actually based on look, those are like distance, but they're all distances on free, one drug, another drug, another drug, a peptide. I don't know, do you remember John Louis? All right, great guy. John Louis' little peptide. Eventually, we used our real substrate. But what you can see, and this is really great, we can distinguish drug binding events, not just drug if it's on or off. We can distinguish, based on our distance distribution, which drug bound and how high the affinity is for each individual drug. Please pay attention. Here we have a broad distribution. That arranges because the rotamers are really assuming a lot of space. Now we have our drug come in, DRV. DRV binds to protease, but not just that. It will displace the paramagnetic labels. It will go in the room and literally pushes them away. And by doing so, the labels will assume the closest possible lowest energy state, as shown here, which is different from DMP, which is also different from RPB, and which certainly is different from S4, which is a peptide itself. And if we now go just for kicks and giggles to our reverse transcriptase, it has actually a real protein in there. Again, that is different. So we simply just developed, in a way, unknowingly, a detection mechanism for how HIV protease actually interacts with the drug. And this is very fascinating. Thank you. And actually, that's based on Dennis's suggestions. Great guy. Huh? It is very, very, very common, very. Yes. Anybody else? Quick question. So who of you guys is going to use ESR for the future of their study? Great. Well, I could say get more people having HIV, but let's don't do that.
Let's thank our speaker.
Thank you very much. Boris, do you have a minute?